Okay, good. Good morning, everyone, for this distinguished webinar series in AI and cybersecurity. Today's speaker is Dr. Nadia Shakoor. The title for today's talk is Advancing Crop Improvement from the Ground Up with the Machine Learning-Driven Analysis of Root Systems and Soil Properties. Dr. Nadia is an assistant member and principal investigator at the Donald Danforth Plant Science Center. She earned degrees from the University of Illinois, University of Chicago, and University of South Carolina, focusing on sorghum functional genomics. Since joining the Danforth Center in 2014, she has directed major projects, including the Sorghum Genomics Toolbox funded by the Gates Foundation and the TERRA-REF project, which has deployed the largest field analytics robot for crop analysis. Dr. Shakoor is also the founder of Agrela Ecosystems, where she developed the PheNode, a field sensor platform for real-time agriculture data. Currently, she leads the USDA-funded FieldDock project to enhance crop phenotyping through the integrated drone and sensor networks and collaborates with the Salk Institute on optimizing carbon capture in sorghum to support climate-smart practices. Dr. Shakoor, it's a great honor to have you as part of the webinar series at UND. The floor is yours.
Great, thank you. I think, there we go. This screen. All right. First of all, thank you so much for the invitation to give a talk today. I'm really excited to be here today and looking forward to some discussions afterward. So thank you for the lovely introduction. My name is Nadia Shakoor. I'm an assistant member and principal investigator at the Danforth Plant Science Center. It's located in St. Louis, Missouri. It's the world's largest plant science institute. And so there's a lot of different programs, and I'm going to tell you a little bit today about my program and some of the ways we integrate machine learning and AI, and hopefully moving forward, we'll continue to expand on that. So first off, happy Halloween to everyone. And so I appreciate you guys joining today. So I want to first give credit to all the folks in the lab. I actually have a very interdisciplinary lab. Because it's a plant science, the goal is crop improvement, plant science. We have biologists, plant scientists, as well as computer scientists and engineers that all work together to kind of get to the projects and the goals that we want to get to. And so having an integrated team is very key. And then we also do a lot of field data collection. So lots of students and part-time folks working in the field to collect ground truth data for a lot of our kind of computation, building our computational models. And so here's the crew here. Mentioned sorghum. Maybe some of you aren't too familiar with sorghum, but this is the crop of interest that my lab primarily focuses on. And sorghum, if you're not familiar with it, is actually one of the most important grain crops in the world. It's the fifth most important grain crops. In terms of acreage, it's a really interesting crop for future climate. It requires less water and can withstand considerable climatic changes. Due to its exceptional drought tolerance, it's the predominant cereal grain in semi-arid regions of the world. And every part of the plant can be used, from the stalk to the grains to the leaves, depending on the usage. There's five major sort of sorghum types that are utilized for different end uses. Grain types for grain, food and feed, bioenergy for making biofuels, sweet sorghums for sugar, forage for feeding cattle and other sort of forage applications, and then cellulosic as well. One of the really interesting things, and what my group is focused on at the moment, is looking at not just the above ground phenotypic diversity, but below ground as well. So sorghum, it's a grass. It has extensive deep roots and very prolific and diverse root exudation profiles. So the roots themselves will exude compounds into the ground. And so this is very interesting from the carbon sequestration story. So the goals of the lab in general are to really trying to combine genetic analysis with high throughput phenotyping to identify and develop sorghum varieties that are better able to capture and store carbon, as well as identify the genetic elements that are controlling these traits in these kind of elite sorghum varieties that we're interested in. So what are the traits we're going after? It's pretty conceptually, it's very straightforward. We're looking for bigger root systems, so having more carbon in the ground, deeper root systems, so having carbon that is as functionally just kind of below the kind of the turnover area, and so just having deeper roots. And then roots that break down more slowly. So having roots that have compositionally have carbon-rich polymers that break down more slowly. So keeping carbon in the ground for longer. Another aspect that my lab is working on is root systems that promote soil health. So looking at those root exudation profiles I mentioned, nutrients in the rhizosphere, as well as the microbiome. I won't talk too much about this today, but if you're interested in that, just let me know and we can have a chat about it. So this is the goal: bigger, deeper roots and roots that break down more slowly for the whole agriculture carbon capture based story that we're trying to develop. So I do want to first acknowledge all the partners in this program. So the project is primarily funded through the Harnessing Plants Initiative at the Salk Institute. That they have a very large kind of agriculture-based carbon capture program looking at different crops. At the Danforth Plant Science Center in St. Louis, we're working on sorghum specifically. And I'm going to tell you all about our program. Have some collaborations with Clemson University. We have a field site out there, University of Missouri, the Land Institute. And then also very importantly, we have pretty close collaborations with the national sorghum producers. So these are the folks that represent the farmers that grow sorghum. And so, and related to that, we have collaborations with all of the major seed companies that sell sorghum here in the United States, and actually globally as well. So these are 6 of the major sorghum seed companies. These folks here, they represent the farmers that grow sorghum. So that's a big part of our work, is trying to identify varieties that farmers are going to want to eventually grow as part of their farming operations. So how do we go about this? It is a huge data collection process. So here in Missouri, we have a field site just about 20 miles from here, in which we grow sorghum. And we grow lots of it, lots of different types of sorghum. So we have, in this particular field trial, we have 344 different varieties of sorghum. These are all in 3 replicates and they're randomized. So, and these are large plots. So for scale, this is a drone image. Each one of these plots is 20 foot by 20 foot. So it's 8 rows, and they're 20 foot by 20 foot squares. So that's quite large plots per variety of sorghum. And then and the reason for that is we do remote sensing of these plots. So we fly drones over these plots. We also are looking at satellite data as well. So trying to get as much resolution as we possibly can in this diversity trial. In these different varieties, we have everything that is planted here in this field has been sequenced, so we have genetic information available for it, and then we're trying to capture the above ground and below ground variation as well to try and identify those genetic elements that might be controlling some of those traits that we're interested in, going back to those bigger, deeper root systems. We also have, I mentioned, we have these collaborating companies that we work with. So we have commercial hybrids, so sorghum varieties that are already in the market that we're testing as well to see if, the whole plant breeding process pipeline can take quite a while, but to see, is there already varieties in the market currently that have those bigger, deeper roots and they're already kind of one step ahead and can maybe even market those varieties as having the ability to potentially capture more carbon. That's sort of the goal with the sorghum hybrid trial that's kind of interspersed in here as well. So this exact same trial that you're seeing here in this image is replicated in two locations. So here in Missouri, as well as in South Carolina with Clemson University. And so the reason for having those dual sites is here in Missouri, we have very rich soils, carbon-rich soils, and in South Carolina at the Clemson Field Research Station, the soil is quite carbon poor and quite poor. It's basically like sand. So having those contrasting environments is important because one of the goals of this project, so we're planting the same varieties of sorghum in the same location for 5 years, and we want to track at the end of the project, has there been any impact of genetics on carbon, nutrients, and other things in the soil? And so in Missouri, where there's already a lot of carbon, maybe that signal could get masked. Maybe in South Carolina, we might be able to detect more changes. That remains to be seen. We're just now in year 3 of the project. So I'm hoping in a couple of years, I might have a good story to tell you along those lines. Another project we have in St. Louis on the other side of our farm is a program that's been sponsored by the National Sorghum Producers. So again, these are the folks that represent the sorghum farmers. And they received a large climate smart commodities grant through the USDA. And we have a small research component as part of that larger program. And what in this particular trial, this one's kind of a fun one. We have 2 different commercial varieties of sorghum. These are commercial hybrids, and then a corn variety. And then if you can see, there's actually 8 of these fields. Each one of these subfields has a different management practice. So the goal for this particular experiment is to see how do different management practices impact the soil properties. So we have tillage, so till versus no till. We have precision nitrogen, and by that I mean we go in at the beginning of the field season, we take a soil sample, and then apply nitrogen to get to a target yield. And then also one of the third variable is a cover crop in which we just planted last week. It's hairy vetch, a leguminous or a legume cover crop. And again, this is a long-term 5-year experiment. We're planting the same commercial hybrids, and then again, seeing the impact of the management trials on those traits that we're interested in. Again, going back to the bigger, deeper roots and the soil health properties as well. In this particular trial, we of course fly the drone, we do the satellite, but we also have sensors in this field. So we have soil moisture, soil temperature sensors in every single plot. So again, trying to see how do soil properties kind of evolve throughout the course of the field season, and then spatially through time as we kind of repeat the same management practices year on year. And so how do we collect our ground truth data? So this is all of the main method that we use to collect our ground truth data is something called soil coring. It's very much exactly how it sounds. We go in next to the stalk of the plant, and we drill down 1.5 meters with this hydraulic drill that's got this clear plastic tube that in this metal casing, we drill down 1.5 meters and we pull out a core of soil. This core of soil is 3 inches in diameter, 1.5 meters. And then students and interns in the lab, what we do next is we take this 5-foot core and then we section it into subsections. Each subsection is then a sample is taken out for microbiome analysis and frozen. A sample, some of it, we take a sample out for compositional testing. And then we also will look and see how much roots are in each of the subsections. So we'll actually wash the soil away from the roots and then dry and weigh the roots as well. Because we want to see, again, how deep do the roots go across all of our different varieties and across our different management practices. So very time consuming, but the goal of this type of data collection is to really get at things like on the right side, you can see we have there's always, in any given field site, there's always small microenvironments. There'll be a sandy patch in your field, or there'll be a dip in the field that really will impact how the crop yields and other traits that it has. So being able to quantify kind of the spatial variation in each plot is something that we're very interested in, even at one single field level. So creating kind of like these heat maps for different traits. And then for the soil coring, what we're trying to get at is, for example, here on the left side, you can see, so in the orangish reddish color, the above ground, so this is the above ground trait. So in this case, it's plant height, and you can see the different plant heights for these different varieties of sorghum. And then this is the below ground. So this is in each of those depths that we cut from the big core, how much roots did we find? And the goal of this is, we want to have bigger, deeper roots. And so what we're trying to find is something like this particular variety. So this is a sorghum variety called Atlas. And in all three of our randomized replicates in the field, regardless of the microclimate impact, you can see that there is this expansion of roots at the 30 to 60 centimeter depth. And what ideally we can find something like this a little bit further down. And this is just to kind of give you an idea of what we're trying to find and why we're doing the soil coring. As you can imagine, the soil coring is quite time consuming and, error prone because, these are human beings washing the soil away from the roots. And if, you don't capture the really, really fine roots, then there's always a human error involved in that. So a method that we've implemented this year, I'll talk about that here in a minute. This is just to show you just the level of variation in the below ground roots, just to kind of convince you that this is a decent approach to go about this type of analysis. So here are all of our commercial grain sorghum hybrids that are currently in the market. So out of those 56 that I had mentioned earlier, you can see above ground grain sorghum. Most grain sorghums kind of look alike, and you can see in terms of the plant height, they're more or less kind of in the same range. But you can see there's quite a bit of below ground variation. So this is looking at the same variety for a couple of years, and you can see that there's some where they're very shallow roots, but below ground, it's a different story. They're going down the full 1.5 meter and potentially even having a good proportion of roots down at the lower depths as well. So we're looking at this in grain sorghums. I mentioned there's other types of sorghum. So these are the commercial forage sorghums, as well as silage and Sudan grass, which is also used kind of for feed and hay. You can see there's a lot of variation across the lines for below ground rooting depth. And so this is just kind of an image of kind of the scale at which we operate. Like we are, this year, we've taken over 1,000 cores, both in Missouri and in South Carolina. It's a big process. We rent freezer units to store the samples because when you pull them from the field, if we're going to do microbiome analysis, we want to keep the samples frozen immediately after we take them from the field. I just kind of wanted to highlight what this kind of data collection in the field really looks like. And then these are some folks washing the soil away from the roots and going in with tweezers and pulling out the roots that they're finding in each of the little subcores. We do above ground phenotyping as well. So I mentioned, we want to try and capture varieties and identify varieties and genetics that are going to be relevant to a farmer. It has to be economically viable just because some plants are some sorghums are very, very tall and they have very deep roots, but that's not necessarily a variety that a farmer is going to want to grow. So we do a lot of above ground phenotyping as well and data collection. We're developing apps for in-field data collection and then using drones and other remote sensing methods to try and quantify and then, of course, manual measurements as well to try and quantify some of these traits in the field that, again, is going to be relevant for a breeder, for the farmer eventually. So this year, what we did, so I mentioned the soil coring is our main bread and butter of how we look at the soil and the rooting depth. Uh, the problem with that is it's error prone because, I mentioned about the humans, they're picking out the roots and we don't really, there can always be errors there. And so this year we implemented something called mini rhizotrons in our field. And so using essentially the same exact type of device that we use to do the cores, we actually pull out the soil core and then insert a clear plastic tube at an angle next to the plants. And then periodically we will go out and take photographs. We'll drop a camera down into this clear plastic tube and then take photographs up the length of the tube. And so you can see here at these top 3 depths, we can see what the root systems look like. So we get a really nice picture of kind of a quick snapshot of how dense the roots are, what color they are, how quickly, if we go at a regular basis. In our management trial, we were actually going out every 2 weeks to see how quickly the roots got down into the lower depths. And so this is kind of another different method that we've been using. And here's where we're starting to apply machine learning methods that I'm going to talk about here in a second. So that was kind of to orient you to kind of our whole program, essentially. But here, so I'm in this image here, you can see the root images are really nice. They're very clear. Unfortunately, most of the time, the root images actually look a lot like this. Those clear plastic tubes will get condensation in the tubes. There'll be sticks, there'll be twigs, a lot of noise in the tubes. And so the images will look like this. And so we're trying to quantify how many roots there are, how big the roots are, and other things. And so Noor in the lab, as well as C4 in the lab, they developed a neural network to try and segment out the roots versus the background. So here is the original image. In the middle column is the image that the student went in and very carefully tried to trace the roots that they could see in the image to train the model. And then in the right is the predicted root systems. And we're actually, in this particular study, we got very, very high accuracy. You can see actually in the lowest row here, you can see these small tiny roots that the model picked up. The student didn't label them, but if you look really closely, they're there. So in some cases, the model actually performed better than the human labeler. So this has been a really, we did this is from last year. And so we were very excited about these results. But then when we tried using the same model, and I think this is the issue that a lot of people face, is the model won't work for a whole variety of host of reasons. And so this was how well it worked last year. And then when we did use the same model this year, you can see it didn't work well at all. And so one of the projects in the lab is we're developing sort of this in-field. So we're deploying the mini rhizotron. All of the imagery is being collected in the field, sent to an edge computer where this is being processed and sent to different types of models. And so, so this same image down here at the bottom, we're adding more models into we have actually like a web-based interface where the images get loaded, and then you can select different models. And you can see we've added so far a couple other models that seem to be working really well for this year. So the idea is to get almost a real-time sort of analysis happening every time we collect the mini rhizotron images, because one of the major issues with this type of work, especially in a field setting, is the time from data collection to analysis to interpretation is just becomes too long to try and make decisions for the following field season. So this is our attempt to try and make this as real time as possible. So this is just another image of those models working a lot better for this year's data collection. And then here is that camera, just kind of show you, this is the Align Scanner. This is the company that actually builds these types of, one of the companies that builds these types of mini rhizotron cameras, and just kind of an image from the company showing you, how it's inserted into the field or into the tube. So I also mentioned that we do sensor collection and data collection from the field. So, there's a lot of environmental variation, particularly in our different field locations and in individual plots. And so several years ago, I started a company out of the Danforth Center, a spin out, in which, one of our kind of major bottlenecks at the Danforth Center, or actually just with our field trials was, all of our field trials are located in different locations, and then going out and being able to collect environmental data at some sort of regular interval, and not. But have data that was high quality enough to start using in different types of environmental models and modeling kind of platforms and systems. And so that's how we started working on building the PheNode. So that's what this is right here on the right side. Basically, it's a remote field environmental sensor, weather station, essentially, but it's been designed for research. So, like from my perspective as a researcher, what are the what's the temporal resolution we need? What is the sensor resolution we need to get really good data? So these systems are equipped with all of your environmental measurements, your atmospheric measurements, soil measurements. So there's soil probes that go down so we can capture things like soil temperature, soil moisture. There's a camera, so having the ability to do it's a low resolution camera, but having that kind of eyes on the field is quite nice, and we can do time lapses and other sort of small scale analyses as well. And then the wireless sensor. Again, these are collecting data multiple times within an hour, but then sending the data once an hour, which is kind of the sweet spot that we found is pretty good for this particular platform. One of the things that we've worked hard to develop, I mentioned in each of our management trials, we have this wireless sensor that's equipped with soil moisture and soil temperature at a couple different depths. We're already seeing variations in soil temperature between sorghum and corn, and we really want to kind of document and see that those soil changes over time, and having a platform like this has been really, really critical. We already have thousands of data points that we're using for our modeling strategies. And this is sort of what that interface looks like for the PheNode system. You can see, wind events. If there's a really strong wind event and your plants fall over, it's all being captured by this system. Again, pretty critical. And it all goes to a standardized CSV that then can be pulled into any sort of data analytics type a system. And so in my research program, we're heavily using these systems to capture high resolution information about the environment in which our plants are growing. So from and this is just kind of an example, like all of the folks that are using the PheNode and where they're located, and for different applications, from monitoring alongside of highways to look at soil, water properties and things, and retention alongside alongside the highways, as well as other applications in different field sites. Out of the PheNode project, so this started out as a technology at the Danforth Center. It was something, it was my postdoc project, and we kind of spun it out into this commercial product. The one sort of limitation with the PheNode is, it's a static system. And then even though the wireless sensors allow you to scale out and to capture a broader area, one of the projects that I proposed a few years ago to the NSF and USDA was something called the FieldDock. So that takes all of the learnings that we learned from this PheNode system, kind of how do you build and operate something that works outdoors in the middle of nowhere, all on solar power. And we've built this other platform now called the FieldDock. So this has been a USDA NSF funded project, and basically it's a cyber physical system that has the PheNode as the brains. But then it also has this edge computing kind of autonomous drone platform. So what the way the system operates, again, for our field trials, is we've developed a GUI, much like the one I just showed you for the PheNode, where you can go in and plan your mission in the field. the FieldDock remotely will orchestrate the mission. The drone will take off. It'll check the PheNode will check the weather. So the weather station will check the weather, make sure you're clear to fly, and then the drone will take off. The drone goes out, collects data over your field site, and then it comes back to the FieldDock. We have postdoc in the lab has been working very hard on the precision landing aspect of it, and he's nailed it. And so it lands on the charging pad. So these are charging panels from SkyCharge, a company out of Italy, I believe. I might be wrong on that one, but out of Europe. So they have a sky charging conductive tiles in which when the drone lands, it'll charge up the drone battery as well. And so the drone lands, it transmits its data wirelessly to the edge computer. And on the edge, we've developed the full end-to-end using open source kind of pipelines that are available, end-to-end image processing pipeline that will take, in our case, the diversity field. So this is all of those 344 different varieties. Process those images and get plot level values, like in our case, vegetation indices. So how much, for example, how green is this plot versus how green is that plot? And that can be, for example, a measure of health. And so this is all done automatically on the edge. We've kind of built the scripts to do that. And then what the FieldDock kind of system does, again, on the edge, it sends up a CSV to the cloud of all of the plot level indices. And then in the cloud, we run something called genome-wide association analysis. This is a type of a model that basically will, because everything in our field we have genetic information for, it will correlate the genetic information we have with the phenotypic value that was captured by the drone and try to associate what are the regions in the genome that are associated with the trait that we're seeing in the field. So, for example, greenness values. What are some of the genetic elements controlling greenness values? And this is an output from the FieldDock platform. So here, this is this is from this past field season. And you can see here is, we flew the drone every week. So the autonomous drone went out every week. And you can see these are all of the values that were captured from our big diversity trial. And so for every single plot, all of the different values at the different dates we collected. And this particular index, this is a vegetation index. So the love kind of looking at the measure of greenness, and you can see as the plants grow throughout the field season, that level goes up. And then here on the right side is the genetic regions that are associated with this particular phenotype, NDVI, at each of the time points. So we can try to pinpoint the genes that are going to be important for this particular trait. And so now we've got this very nice sort of automated platform that we're extending out, not just for the above ground traits, but also now for the below ground traits. This is a water index down here. So you can see it kind of goes down as the field season progresses. And so we're developing models to look at using the same sort of system, and also our soil coring data to see, can we predict things like soil nitrate and soil organic carbon. And so this is just sort of early work by Babukar in the lab. He's a postdoc who's been trying to develop models to predict these traits. Using our ground truth data that we send to the lab from the drone data specifically. So using the features from the indices collected from the drone to try and develop models for predicting things like soil organic carbon and nitrate. And so he's getting pretty good results in the top depths. Lower depths, we're still kind of thinking about strategies on how to look at those traits a little bit further deeper down into the soil. One thing that we have done in order to look at things like carbon further down in the soil is an indoor hyperspectral system, which I'll talk about here in a second. But this slide here is just more work from Babukar showing, again, from the drone, kind of what the images look like, and then trying to predict the different traits versus our measured values, predicting biomass, above ground biomass as well. And then we're also seeing how different models perform differently for different genotypes. And that's an open question in our lab that we're really trying to understand, why do certain models work a lot better for particular genetics. So not sure if you have any thoughts on that. Would love to chat about it. But this is more work from Babukar in terms of the aerial imaging to predict traits in both above and below ground in our fields. With Bishal Roy, he's a graduate student at St. Louis University under the supervision of Fawcett Sagan. I'm one of his co-mentors, and so we've developed, so he's been very successful in developing an indoor hyperspectral pipeline for quantifying soil organic carbon here specifically, and now he's working towards other traits as well. So using all of the different the soil samples that we took from each of the different depths of the soil core, we dry down those soils, mill them, and put them on a 48 well plate. These plates then are run through this indoor hyperspectral scanning platform. And so he's been working as part of his PhD project to try and identify what are spectra that are going to be indicative or for estimating soil organic carbon. And so he's seen some really good kind of correlations in the SWIR region, very, very high, well, actually quite high values in trying to predict soil organic carbon. This was from a relatively small data set. So this year we're expanding it and hopefully kind of trying to improve these accuracies, but then also try to really hone in on the regions that are going to be useful for those particular traits. Here at Soil Organic Carbon, I'm really interested in this particular technology for other soil traits as well, because sending samples off to the lab for wet lab analysis is always quite expensive. So having a high throughput method, even though it's, still, it's not thousands of samples, but it's still very high throughput, much more cost effective to do kind of field level analysis at like every single genotype every single year. This kind of gives us the ability to maybe start thinking along those lines. And so with that, we do a lot of indoor work. I didn't mention that today, but I will leave it at this. There's always, like I mentioned, lots of students. This is so much labor to capture all of this data. So a lot of students from the Danforth Center have come through my lab over the last few years, all of our collaborators and folks that work on these projects, and then of course the funders as well. So be happy to take any questions.
That's awesome, Nadia. Thank you so much for your fascinating presentation. So we'll open up for Q&A and we'll get the Q&A coming in on chat. But maybe I'll start with some question. I think, in terms of sorghum biomass predictions that you talked about, do you also overlay other external variables, other environmental variables other than the ground truth? I mean, I'm just curious to see, what is the number of variables that you're dealing with, and what are the variables played an influential role in the biomass prediction?
Yeah, that is a great question. And that is why I'm such a big fan of the environmental monitoring, the real-time environmental monitoring. So each of our, I mentioned the PheNode, but we've deployed in those two different field sites, we have about 10 PheNodes and about 60 plus of those small wireless sensors to capture exactly what you just said. What is the local environment over the course of the field season? Because that's going to impact your biomass yield or grain yield, whatever yield you're going after. So we very much want to pull in the environmental data as well as we're developing this. So that's something that Babukar is working on as well. So.
Super. And then you talked about your company PheNode, and how many, I mean, do you have, I mean, is it just starter? Do you have how many weather stations that you have? I'm just curious because we've been working with UCAR, the NCAR's National Lab, and on trying to see whether, that they have weather station that they put in low income countries and try to see whether we can and help them to provide some kind of climatic predictions on events. But do these weather stations, what you have, can talk to each other, or they are just deployed and provide a standalone thing on Shen? How can you share some insights on this phenomenon?
Absolutely. Yeah. And I'm happy to maybe do a demo offline at some point. But yeah, so all of the So you, as a user, you would have, so in my case, I have all of my phenodes at all of my different field sites, and I can remote into each one and then go in and set, oh, I want data from the last 6 months for XYZ sensors, and you can download that data directly from the GUI, and all of it comes out as a CSV. So that's kind of the goal of it. So yeah, so it's all connected in the sense that it's all comes to one user dashboard, and you can scroll through all of your um, kind
So,
of the systems that you've deployed. So yeah, very
okay,
much.
so when of course when we talk about these weather stations, there's a lot of them on the market. How
Yeah,
cost effective is this one, and for a scalable solution, I was wondering, can you shed
yeah,
some light on more on the weather station? Because we have some interest in see how we can securely transport the weather station data between these stations, and we've been working on USDA and a bunch of other agencies to see what we can able to do. So maybe I see some synergy from what you guys do. Maybe we'll find ways to
absolutely.
collaborate.
Yeah, so the main way that so our sorry, for some reason this is, let me see if I can. Pick up that or sharing maybe the wrong screen. Yeah, so our sensors, it really depends on which sensors you want on the system, right? So it starts at about 3,500, which is very cost competitive compared to others. But then it kind of goes up from there. If you want to say a really cool multispectral camera, or you want you know, the cost of the system depends on the sensor you want. So the idea behind the PheNode was to have like a plug and play platform. So having the option of doing, do you want soil moisture, soil temperature, or do you want the fancy $1,500 soil probe that goes down and takes readings at multiple depths? So it's designed for research and the research sort of application. So um, but yeah, they start at about 3,500. That's sort of the base level, and then it goes up from there, which is again very cost competitive with everything else on the market. And I do want to emphasize this was very much designed from my perspective as a researcher. So that's kind of, I think, the differentiator, because it's the quality that I want and expect, and from a sensor platform. So.
How much wind speed that they can stand? Are they sturdy? Because this is something that we've been talking when you want to deploy these things in real ag fields, and do they really have that much of endurance to withstand?
Yeah, absolutely. So we've had these systems in the field for well over 2 years. They've been running continuously. So we have well over 1 million data points from the system. And we actually use the wind. So in one of our field trials, this is just kind of a really good story. In one of our field trials, we had it was actually a collaborator in which he had modified a plant so it stood upright. And so generally the plant falls over. He got it upright, but the plants in the field fell over. But we had a really high wind event. And so the PheNode clocked 36 miles an hour at that particular, because at the airport where you usually get your weather station data, it was saying the average for the hour was 18 miles an hour. But it gusted to 35 miles an hour. And so the PheNode captured that, and he was able to show his funders that, hey, it wasn't my plant, that it was just the wind speed was just so high. So we've seen well over 40, 50 miles per hour winds, and the PheNode stays very stable. We've never once had issues of it falling over or dislodging. So there's the main PheNode tower. There's also these little loops on the top of the PheNode where you can install guy wires to anchor it into the ground. In Georgia, we have it alongside the highways, so there's a lot of wind and you know, sort of so.
Sure. And you mentioned in one of those other slides, Sharp DNA and Sharp RFR, the nice newspaper and the new paper from whoever the authors are. Are those XAA techniques that Sharp and Lime, that in hybrid combination of DNA and RFR, or is this the name Sharp? Do you know what those methods?
Yeah, I would have to ask our the folks that work on it. Again, I'm a computer science or I'm a plant scientist that really appreciates how computer science works. So I would
No
have to,
worries, no worries. I think that's
yeah,
good. We can even read through the paper. I think
yeah,
that's no problem.
absolutely.
Okay.
Yeah.
And
Here, I
uh,
think it just logged in, but here's kind of an example of what our interface looks like. So I just logged in, and so you could, so these are all of our phenodes that are currently deployed right now. Field season's ending, so a bunch of them are offline, but this one, for example, is our field research site here in St. Louis. So you can see what time the last measurement was taken about 30 minutes ago here locally.
mm,
Kind of what the, you can go in and say, oh, hey, what's happened for the last 90 days, for example, and you'll see, oh, hey, on this particular date, the wind gusted quite a bit, 16 and a half miles an hour. So this is kind of the user experience I want for the researcher to have all of this data just right there at your kind of command. And so, for example, here's one that I know has a camera system installed in it. So let's see, does it have, maybe it'll load up, maybe not this one. Let's see if there should be, but yeah, there's all sorts of really cool kind of aspects to this. And then how you can go in and download the data, adjust your data download preferences, and get all of your real-time data in a CSV. So, and again,
okay.
happy to do a proper demo. And so, yeah, for you. Yeah,
Uh, okay. Well, I'll see whether we have any other questions. Maybe I'll share something once. And I just want to okay, let me see if I can. You heard of 3D Pause weather station?
no, I don't think so. Is that,
Okay.
yeah.
Let me see. Do you see the website what I'm having?
Oh yeah, that's cool. Yeah, yeah,
So
that looks
this
nice.
is UCARS, which is your an University Consortium Atmospheric Research, part of NCAR. And I've been
Okay.
working with Paul Snyder, who is the assistant director there, and he is wanting a low cost. I think it costs about 300 bucks, and a very
Yeah,
scalable, very cheap, and at the same time easy to teach and train these third world countries and also people in rural areas, tribal areas where we can able to deploy quick and train them. So, but I know that yours is more research, more into the piece,
yeah,
but maybe we'll find a way to connect the work what you're
absolutely.
doing, so
Yeah, what we found is, that's always there's that cost kind of data quality exchange, right? Sensors very much when you get to the very, very low cost sensors, the quality of the data gets quite poor, and less reliable. And so having, again, for research, for models, I am a big proponent for not having the most expensive sensors, but not those very, very cheap sensors that will actually stop working. That's another thing that with a lot of these low cost systems, as long as you have it at a university or somewhere where people can go out and swap things, if you have something in the middle of nowhere for months at a time, those sort of cheaper options will fail. Like I mentioned, we've been at it for 10 years. We've had so many iterations, and we've really found there's a sweet spot in in cost.
so do these weather stations operate on Wi-Fi or do they on cellular network or what?
Yep.
How do
Both cell network, a Wi-Fi if you have it, and so but primarily cell network. The wireless sensors are LoRa, and so
LoRaWAN? Okay, yeah, yeah, okay.
yeah, the wireless sensors operate via LoRaWAN. The phenote itself communicates primarily through cell, but if you're next to, again, if you're at a university or somewhere where there's a good Wi-Fi signal, then that's
One
perfect.
one other final questions. We also got new funding from NSF on establishing an IUCRC Center for AI and
Oh,
for critical
congrats.
infrastructure.
Yeah.
So part of that is I'm doing a planning workshop sometime in spring, looking at ag and energy, a bunch of other domain too. So maybe I'll send you some invitation for that when we have the date for spring and and maybe your center or maybe PheNode could be part of this IUCRC. Have you heard of a IUCRC
I
program?
have, yes. Yeah,
Okay,
yeah. I've
so
heard about those programs. Very competitive, so congrats on getting it.
yeah, so maybe that's something interests you or your company, we'll talk and I'll send you some more information about that. Yeah.
Absolutely. Yeah, that would be cool.
Super. Thank you so much for this morning. And I think you will receive a nice flag and letter from our dean and appreciate for
Oh,
taking the do this webinar. And I think I look forward to collaborate with you as we work on more projects.
oh, that would be wonderful. Yeah, I do too. Thank you so much. Yeah.
Thank you.
Have a great rest of your day.
Thank you.